schematic representation of oogenesis. Okay. Oogenesis is defined as the process of production of haploid ova from diploid gonal cells through meiosis in ovary is called oogenesis. Oogenesis occurs in ovarian follicles of ovary. The ovary contain the primordial germ cells. These are called primordial germ cells. Primordial germ cells or egg mother cells, which are diploid in nature with 44 autosomes plus a pair of 6 chromosome denoted by XX in female, that is 2n is equal to 46. Oogenesis is also divided into 3 steps: multiplication phase, growth phase and maturation phase. During the onset of oogenesis, primordial germ cells lining the ovary undergo multiplication through ordinary mitotic division, divide and redivide to form a large number of cells. The resulting cells are called oogonial cells. The resulting cells are called oogonial cells. Oogonial cells, which are generally diploid in nature. Primordial germ cells, that is 25 weeks of intrauterine life. This is generally primordial germ cells undergo multiplication during what is called as the development of embryo. 24 weeks of intrauterine life or embryo, and that oogonial cells are formed generally, that is nothing but at the time of what is called as the growth of embryo, growth of embryo, growth of embryo, embryo, that is before puberty, before puberty, that is during childhood. So this constitute multiplication. After attaining large number, multiplication stops and enters into growth phase. Growth phase in oogenesis is long compared to spermatogenesis because there is a deposition of reserve food metals in the form of yolk. The process is called vitellogenesis and also involves biological synthesis of biomolecules like proteins, carbohydrates, lipids, etc. The volume of cytoplasm increases and the resulting cell is called primary oocyte. Primary oocyte, which is also diploid in nature with 44 autosome plus XX, which is also diploid. So these are also diploid in nature, diploid. Whereas the primary oocyte, this is after puberty. This is after puberty. Puberty is generally arrested at meiosis 1. Arrested at meiosis 1 of prophase in diplotin, followed by growth of Graafian follicle, and then undergo what is called as the first meiosis. First meiosis. First meiosis is a reductional division where the chromosome number is reduced to half. So here the events like synapsis, tetrad formation, crossing over occurs after these events, where the primary oocyte divide unequally to produce larger secondary oocyte and smaller first polar body. Secondary oocytes are structurally dissimilar but genetically similar, but there is unequal division. Only thing is to eliminate unwanted chromosomes, but unequal distribution of cytoplasm to produce only one ovum per menstrual cycle. And this secondary oocyte is generally undergo second meiosis. In second meiosis, it is an equational division, once again divide unequally, and this is generally arrested at meiosis to half metaphase, arrested at metaphase 2, and then secondary oocyte is released into what is called the abdominal cavity, from where it enters into fallopian tube. After entering into fallopian tube, entry of the sperm, then it second meiosis completes before fusion of male nucleus with female nucleus, and once again divide unequally to produce larger ooted or ovum and second polar body. In majority of the vertebrates, including humans, where the first polar body undergo disintegration, or it will divide into produce two polar bodies in other animals, but in humans and vertebrates, the first polar body do not undergo second meiosis, it undergo disintegration. So in all animals, 3 polar bodies are produced at the end of oogenesis, and only one ooted or one ovum is produced, and whereas in humans only 2 polar bodies are produced and one ovum is produced, and this ooted or ovum can directly take part in reproduction without undergoing any change. So this is about what is called the oogenesis. This is called maturation phase. Maturation phase is also called as the meiotic divisions, and this is called multiplication phase. Multiplication phase. So now let us study oogenesis. Oogenesis is defined as the process of production of haploid ova from diploid oogonial cells through meiosis in ovary is called oogenesis. And whereas oogenesis occur in seminiferous ovarian follicles, of ovary. Oogenesis is also divided into 3 steps, namely 1. Multiplication phase. Second one is called growth phase. Third one is called maturation phase. These are the 3 steps or phases involved in oogenesis. So this process of production of haploid ova from diploid oogonial cells through meiosis in ovary is called oogenesis, and it occurs in ovarian follicles of ovary, and it is divided into 3 steps, namely multiplication phase, growth phase, and maturation phase. Multiplication phase During the onset of oogenesis, primordial germ cells or or egg mother cells lining ovary undergo multiplication through ordinary mitotic division, divide and redivide to produce large number of cells called oogonial cells, which are also diploid in nature. Multiplication of primordial germ cells occur during development of embryo that is 25th 5 weeks of intrauterine life IUL. After obtaining large number multiplication stops and enters into next phase. The next phase in oogenesis is growth phase. Each oogonial cell increase in size due to synthesis of biomolecules like proteins, carbohydrates, growth phase in oogenesis growth phase is longer due to deposition of reserve food materials in the form of yolk. The process is called, the process is called vitalogenesis, and preparation of cell for deposition of yolk is called pre-vitalogenesis. After pre-vitalogenesis, after growth phase, oogonal cell increase in size, the resulting cell is called primary oocyte, which is also diploid. After attaining maximum size, growth phase stops and enters into next phase. It enters into next phase, that is called maturation phase. Maturation phase involves meiotic divisions. They are first and second meiotic divisions. First meiosis is a reductional division in which chromosome number is reduced to half. The events like synapses, tetrad formation, and crossing over occurs between non-sister chromatids for exchange of genetic material. And arrested at meiosis 1 of prophase, that is in diplotin stage. After this primary oocyte divide unequally to produce to produce larger secondary oocyte and smaller first polar body. So the first maturation phase involves two meiotic divisions, first and second. First meiosis is a reductional division in which the chromosome number is reduced to half. The events like synapsis, tetrad formation and crossing over occurs between non-sister chromatids for the exchange of genetic material, and this is generally arrested at meiosis 1 or prophase in diploid stage. After this primary oocyte divide unequally to produce larger secondary oocyte and smaller first polar body. The secondary oocytes are structurally similar and genetically dissimilar. And genetically similar. The first polar body undergo disintegration in humans and all vertebrates, but in some animals first polar body also completes second meiosis. The secondary oocyte undergo second meiosis and arrested at metaphase. The secondary oocyte once again divide unequally to produce larger oocyte or ovum and smaller second polar body. The secondary oocyte, the egg is released into body cavity at the secondary oocyte stage. Enters into fallopian tube, sperm enters into ovum, and before fusion of nucleus undergo second meiosis second meiosis to produce ooted and second polar body. Second polar body. So at the end of oogenesis, only one ovum and 3 polar bodies are produced. 3 polar bodies are produced. Ovum or ooted can directly take part in reproduction without undergoing any change, unlike spermatogenesis, unlike spermatids. This is about the oogenesis. The overall schematic representation of oogenesis is here. First thing is primordial germ cells lining the ovary undergo multiplication through ordinary mitotic division, divide and redivide to produce large number of cells called oogonia, and this oogonia is generally produced at embryo stage, that is 25 weeks of intrauterine life. And this oogonia, after obtaining large number, and it enters into next phase called generally growth phase, where the deposition of reserve food materials takes place, biosynthesis of biomolecules, the volume of cytoplasm increases, each oogonia increase in size to form primary oocyte. This is at birth till puberty. So the primary oocyte, after attaining maximum size, undergo first meiosis. You know that first meiosis is a reductional division where the chromosome number is reduced to half, and this is very important phase in the oogenesis, and this is arrested at prophase 1 of diplotin, followed by growth of Graafian follicle. And this Graafian follicle enters into or divides into 2 cells. One is called the first polar body and secondary oocyte, where the first polar body undergo degeneration in humans and all vertebrates, but in some animals even first polar body also completes the second meiosis. The secondary oocyte undergo what is called as the second meiosis. The second meiosis is arrested at metaphase stage, and then followed by release of what is called secondary oocyte into body cavity, and this process is called ovulation. After ovulation, the secondary oocyte enters into fallopian tube and reaches the ampulla of the upper fallopian tube, and again the sperm enters into the ovum, that is called insemination. After entry of the sperm, before fusion of the nucleus, where the secondary oocyte divides into second polar body and ooted or ovum, where this ovum can directly take part in reproduction without undergoing change, unlike what is called as the spermatids. And this ooted undergo what is called as development into embryo if it is fertilized. If it is not fertilized, it undergo degeneration. This is the overall schematic representation of oogenesis. Now, hormonal control of oogenesis. The oogenesis is controlled by the hormones which are generally secreted by pituitary gland under the influence of hypothalamus. Hypothalamus, commonly called as master of master gland or the thermostat of the body. It releases an hormone called GnRH, gonadotropin releasing hormone and stimulates the anterior pituitary gland. Anterior pituitary gland. This anterior pituitary gland releases the hormones like what called as LH and FSH, which stimulate the ovary. The ovary secrete hormones like estrogen and progesterone. Both are for negative feedback mechanism. And this progesterone, it helps in development of what called as corpus luteum. And whereas estrogen, it is for positive feedback mechanism. Generally stimulate the hypothalamus to release GnRH and stimulates pituitary gland, releases LH and FSH, stimulates ovary to produce what called as estrogen and progesterone. And this estrogen is for the growth of what called as ovarian follicles. This is about the hormonal controller of oogenesis. Okay, first one, hypothalamus, the master gland, releases GnRH, gonadotropin releasing hormone, stimulate the anterior pituitary gland, a glandular part. It secretes two hormones, LH and FSH, and which in turn stimulates ovary, and the ovary releases two hormones. Estrogen is a female sex hormone responsible for the growth of what are called the sex organs, even secondary sexual characters, and stimulation of what are called the ovarian follicles to produce ova through oogenesis. And then progesterone, it is called pregnancy hormone, and it helps in it is secreted by corpus luteum. So that is generally it inhibits the secretion of what are called progesterone if the womb is not fertilized. Okay, yes, between spermatogenesis and oogenesis. Differences between spermatogenesis and oogenesis. First column is spermatogenesis and the second column is oogenesis. First difference, spermatogenesis and oogenesis, it involves the production of sperms. Oogenesis, it involves the production of ova. It involves the production of ova. It occurs in seminiferous tubules of testes. And this it occurs in ovarian follicles of ovary. Where spermatogenesis, it is a long process, whereas oogenesis, it is a short process. And again here growth phase is short. Here growth phase is long. Due to Vitellogenesis. Vitellogenesis. And again here first and second meiosis occur within seminiferous tubules, and here first meiosis occur in ovary, and second meiosis occur in fallopian tube. Primary spermatocyte divide unequally. Here primary oocyte divide, primary spermatocyte divide equally, and primary oocyte divide unequally. And again secondary spermatocytes are structurally similar and genetically dissimilar because contain X and Y chromosome. Here secondary oocytes are structurally dissimilar and genetically similar. From each primary spermatocytes, four spermatids are produced. From each oocyte only one ootid is produced. Spermatids undergo spermiogenesis or metamorphosis, and here ootid can directly take part in reproduction. And one more difference, that is at the end of spermatogenesis, four sperms are produced. At the end of oogenesis, only one ovum is produced. And last difference here, polar bodies are not formed. Here three polar bodies are formed. That is about differences between spermatogenesis and oogenesis.